Now, the last part of the chapter, we'll talk about two different bacteria. Those are Yersinia and Haemophilus. So the first one is Yersinia. We're going to be discussing Yersinia pestis. That is the bacteria that causes the plague. It is a gram-negative rod. The virulence factors of the plague, or sorry, of Yersinia pestis, is that they are capsulated, so the capsule and the envelope, both of these protect against phagocytosis. Um, the Yersinia pestis bacteria secretes a coagulase enzyme. Now, coagulase um, causes blood clots, and we'll see how that manifests in the signs and symptoms of the disease. It also protects the bacteria, where the bacteria again surrounds itself with blood clots, hence preventing the immune system from seeing and recognizing the bacteria. It has an endotoxin. The endotoxin, again, is the lipopolysaccharides that are found in all of these gram-negative bacteria. It also secretes this murine toxin. Murine toxin is important in the life cycle. Part of the life cycle are fleas. Okay, fleas don't get sick because of Yersinia pestis, but they do transmit the disease to humans. And the reason that this Yersinia pestis can survive in fleas is because of this murine toxin. So it's a very important part of the life cycle. Okay, so let's talk about the epidemiology and life cycle of the plague. Um, so we, excuse me, we can contract it through our contact either with wild animals, and we call that sylvatic plague, or through domestic animals, and we call that the urban plague, or we can contract it from other humans. Okay, we'll see that Yersinia pestis is found in about 200 different species of different mammals, like rodents and squirrels. Again, it doesn't cause the disease in them, but it is part of the life cycle where eventually we could contract the disease. Okay, so here's a life cycle of Yersinia pestis. So you can see all of these are part of the cycle. There are rats, mice, chipmunk, squirrels, and when they get bit by a flea, the flea or the aerosol from these animals could eventually transmit the infection to humans. And then there's also the human to human infection. So when it's transmitted to humans through these flea bites, we'll see that the bacteria will be transported to the lymph nodes. Okay, so let's say the bite is in the upper limb, the lymph nodes will be affected, will be the axillary, or if the bite was in the lower limb, the infected and affected lymph nodes would be in the inguinal area. Now these lymph nodes are going to will swell up, and they're painful and they're inflamed, and we call that the bubonic plague. So bubonic plague indicates the involvement of and swelling of the lymph nodes. This person is now contagious, and through the droplets could affect another person. This person will not get the bubonic plague, though, because there are no lymph nodes. There's no bite, there's no flea bite here involved, but they will have what is known as the pneumonic plague. Know if a person is bit by a flea, they can have both bubonic and pneumonic plagues. Okay, so the pathology of the plague, it has a very low infectious dose. Okay, so between 3 to 50 bacteria are needed in order to get a person sick. Okay, so it is a very highly virulent bacteria with a very low infectious dose. So what are the manifestations of the infection? We already talked about the bubonic plague. So there's a little bubo where there's a swelling of the lymph node. In this case, it is the axillary lymph nodes. So it could be either the groin or the axilla. It could also lead to a systemic plague where due to the coagulase enzyme, there are several blood clots. And you can see that it eventually can lead to subcutaneous bleeding. It can lead to gangrene, and that is why it is nicknamed the Black Death. And it can also lead to the pneumonic plague, where the lungs are involved. Okay, and again, this is highly contagious and it is fatal without treatment. So how do we diagnose and treat or even prevent plague? So diagnosis really depends on the history, on some lab findings, taking a little sample off of the buboes, and that would be the specimen, and you would run some tests and diagnose it. It is treated with Cipro, which is an antibiotic. There is a vaccine, it's a killed or attenuated vaccine that is available. Prevention would be to, since the source of the infection are rodents and chipmunks and squirrels, you want to try to control the rodent population among human habitats, so in cities and so on. So that's the plague. In this part, we'll talk about Haemophilus. Haemophilus is a bacteria that loves heme, or it loves blood, so it's called the blood-loving bacilli. These are tiny gram-negative pleomorphic rods. They are sensitive to drying, temperature extremes, and disinfectants. They don't really grow on blood agar, but what they do grow is on chocolate agar. So the difference between blood agar and chocolate agar is that blood agar has, well, blood, while chocolate agar, you take blood and you shake it vigorously, releasing hemoglobin. And that is what gives you that brown color. Okay, so blood has the hemoglobin inside of the red blood cell. That's why you get this red color. While chocolate agar, you've released the hemoglobin out of the red blood cell, and that is why you get this brown color. Okay, so hemolyphus can grow on chocolate agar, not blood agar. Okay, it needs hemoglobin. Okay, now Haemophilus influenzae that can cause pneumonia, it can cause bacterial meningitis, and it could spread through the bloodstream, leading to bacteremia. There is a vaccine against it, against the B serotype of H influenza, because there's an A and a B serotype. The vaccine is against the B serotype only. Haemophilus meningitis, again, it's a bacterial meningitis. It leads to all the signs and symptoms of any kind of meningitis, right? So there's fever, there's vomiting, there's headache, there is photophobia, which is not being able to look into any light, okay? It can lead to stiffness of the neck. There is about a 90% fatality rate if a condition is not diagnosed early and treated with antibiotics. Other non-life threatening conditions that are caused by Haemophilus are conjunctivitis, epiglottitis, cellulitis, sinusitis, otitis media, and arthritis. Another kind of Haemophilus is called Haemophilus ducreyi. So the first Haemophilus we talked about was Haemophilus influenza, and that leads again to pneumonia and bacterial meningitis. Those are the main diseases you want to know for that microbe, while Haemophilus ducreyi leads to chancroid. Now, chancroid is a sexually transmitted infection that leads to these little chancres, kind of look like these ulcers that are found in the groin area. And again, you would want to take a sample and isolate it, look at it. You're going to grow it on a chocolate agar, and if positively identified, you're going to be treating these with an antibiotic. Now, question here, the bubo of the bubonic plague is which one of these? Is it an ulcer where the flea bite occurred? Is it a granuloma in the skin? A granuloma is a little swelling with that looks very irregular. Or is it an enlarged, swollen lymph node? Or is it an infected sebaceous gland? So if you remember from the previous slides, we saw it as an enlarged, swollen lymph node. So if that was your choice, you have answered correctly. And that is the end of this chapter.